Hello, good afternoon. My name is Sonja Loges and I'm head of the Division of Personalized Medical Oncology at the University Hospital in Mannheim, Germany, and at the DKFZ in Heidelberg, Germany. I'm very happy to moderate this state-of-the-art and top-notch webinar. Thanks for coming. The title is Dissecting Clonal Evolution in Cancer Using Personalized Oncology Platforms, which I think is a highly timely topic. And I just want to very briefly introduce the speakers to you. So today's speakers are Michelle Dietzen and Philip Jost.
Thank you very much for your introduction, Sonja. It's a great pleasure to be able to talk today about personalized oncology platforms and how we can use it in the clinical context, and also for instructing research, basic research from clinical personalized oncology platforms. It is a great pleasure to present not only the clinical concept of our molecular tumor board that we had over the last 5 years or so at the Comprehensive Cancer Center in Munich, but also give this talk together with Michelle Dietzen, who's a PhD student, a formidable collaborator on understanding clonal evolution in cancer and clonal evolution that has been developed out of the molecular tumor board concept that we've done in Munich over the last year. So I'll start with about 10 or 15 minutes of introductory slides. I hand over to Michelle in the meantime, and then she'll be handing back for the last few slides to me again, and then we're open for questions. So, sorry. Now, the core question that we, of course, want to answer is, how does personalized oncology work, and how does it have, whether it has any beneficial impact for our patients? Because you can mention personalized oncology in every context in a clinical setting. However, to really make it the long way to an actual benefit in the clinical context it's a large step and a difficult step. And I think we are all challenged by the opportunities that arise from personalized oncology, yet we have to critically ask ourselves where we can have a beneficial impact for our patients. The basic concept, as you know, is a heterogeneous patient population. Now we want to identify common genetic markers, but of course, maybe not only common genetic, but also epigenetic, metabolic, or proteomic factors in the future to really be able to individualize therapy for cancer patients based on molecular markers that we identify. And the core concept, and I'm sure all of you have heard about it, I'm really going through this very quickly just to make sure that we are on the same page here. But NGS-based detection of driver mutations really underlies a core problem, or let's say a core aspect, which is important to understand. You can see here a chart showing total somatic mutations in cancer over the individual hotspot mutations, and you can appreciate that only a few mutations really have a high incidence in cancer overall. This is a pan-cancer setting. Whereas many mutations occur individually with a relatively low incidence, indicating that, of course, despite the fact that those tumors have an individual low incidence mutation, they might still be clearly driven from this mutation, and therefore we need to understand the whole spectrum of mutations in those cancers. And of course, understanding an individual genetic lesion in a certain patient, in a certain cancer tissue, is hampered by the fact that we not only need to understand different mutation in different genes, and as you've seen here, this is the frequency of certain alterations, P53 or KRAS or BRAF being very prevalent mutations, but also by the fact that within individual genes, very different mutations can occur within hotspot mutations. Example shown here is BRAF, V600 mutations. But of course, there are certain other mutations, such as mutations within the same gene, but other locations within the gene, indicating that, of course, we need to understand whether they are activating or inactivating, whether they do have a pathologic consequence or not. And moreover, we need to understand whether individual tumor types really do require mutations, or whether harbor mutations that are pathogenetically important or not. So the frequency, for example, for BRAF V600, the melanoma is very high, but also exists in myeloma and lung or prostate cancer, but a certain to much lower frequency. So we need to understand genes, mutations, variants, and entities to really annotate our data more clinically. And of course, biomarkers are the basic concepts to understand personalized oncology and personalized medicine. This is just an overview that should illustrate that we have a lot of different biomarkers based, of course, on sequencing, but not only sequencing. We have qPCR, array-based analyses. We have classical immunohistochemistry. We know that translocations, RNA-based translocation detection, and even FISH-based translocations can instruct our clinical handling of individual cancer patients. And therefore, we need to really understand for every tumor type, as shown here, for example, breast, skin, or liver, that a plethora of different mutations per tumor are important in standard of care therapy, and of course, then also in future personalized oncology concepts. And as I said before, the levels of molecular analytics in molecular oncology are, of course, based on genomics at the moment. So precision oncology is almost considered to be largely genomics, but we need to incorporate aspects such as epigenomics, transcriptomics, which is more likely the case already, but also proteomics and even metabolomics to really try and stratify patients on a much better level compared to what we have on a purely genetic level. And of course, this gives a very high heterogeneity in data that we need to understand and illustrate and validate basically in a setting where we try to understand functional consequences. We should not just correlate those data, but really need to functionally understand those individual lesions. Now, it's a lot of discussion currently going on how to best annotate our patients in clinical concepts within personalized oncology. And we know that we need decision support systems. Decision support systems are basically concepts and software-based concepts which should integrate general information, as shown here, this upper branch, but also personal information from those patients. So using both genetic data, pharmacogenomics, population information, but also electronic health records, and all of these information that we can get from individual patients, and really try to get to a diagnosis and a prognosis concept with this decision support system. But the question really is, what is this best decision support system? And I do think we do not have the final answer to this at the moment, because a lot of those software tools that are available at the moment provide some benefit, but certainly do not allow us to fully depend on it or rely on it in the clinical setting at the moment. So there's a lot of research going on in this direction. I think it's very important to do this, and it's also very important to develop our concepts in this decision support system further in the future. Now, what we do in our molecular tumor board, we of course utilize our patient data. We perform a pre-processing together with the molecular pathology department. We perform a variant calling, which is basically done mostly by the molecular pathologist in the identifying whether an individual mutation is relevantly frequent within the sample, what the tumor cell number or the ratio of this tumor cells compared to normal cells within the tissue is. We then need to go to variant annotation and use largely in silico-based data bases that allow us to identify pathogenicity, basically using, for example, COSMIC, ClinVar, Genome AD, or Varsome, which are basically software tools, databases to allow us to individually screen individual mutations for their relevance in a clinical context. And then we go to variant interpretation number 4, and this variant interpretation really is the main focus of a clinician with a molecular tumor board interest, and this interpretation is a highly time-consuming and meticulous effort to really try and screen clinical trials, databases for treatment options, but also with the light of toxicity, availability of drugs, and also the actual patient in view of how clinically relevant this could be for the individual patient. And finally, a report is generated that will be then sent forward to the treating physician, who can then instigate the therapy. Now the question is, what have we learned from the initial years of the molecular tumor board at the Comprehensive Cancer Center at the Technical University in Munich? I'd like to give you some clinically relevant, as I think, clinically relevant data from what we've generated over the last years. This is, of course, a snapshot, and more in-depth analysis will be done with longer follow-up. But what I can show you is, at least at the moment, we and I'm trying to click. Okay, sorry. So this pie chart shows you the different disease entities that we have included into our molecular tumor board over the last years. You can see a very balanced inclusion of patients according to different cancer entities, with about 23% of gastrointestinal tumors, 20% of gynecological tumors, 20% of dermatology tumors, and with 50% of sarcomas. We have a very spread and very colorful picture of different disease entities. So it's a very broad, really pan-cancer inclusion. There's no individual disease that is really so much in the forefront that you could think that there's any bias towards the analysis. I mean, breast cancer, melanoma, and sarcoma indicating, of course, with colorectal, the most frequent disease types. Which participating clinical departments did submit their patients to the molecular tumor board at our center? We have, again, a very balanced inclusion from the centers that you can see here: internal oncology, gynecology, urology, gastroenterology, and of course, in red, 20% of partners from external sites that sent their patients and samples also to the molecular tumor board. These are the patient numbers over the last years. You can see a steady increase from 2016, 17, 18, and 19. And in total, we have now had contact with 558 patients until the first half of 2020. And numbers have increased since then, and we're at about 900 patients so far. This is the data that shows you clinical outcome, and it's depicted similar to what you would think in a process where on the left-hand side in this bar is a response prior to therapy, so the best response before entering the molecular tumor board, and on the right-hand side you will see response post-targeted therapy. So that's what we were able to achieve with our molecular tumor board suggestion. And of course, you lose a lot of patients before you can actually enter them into this the treatment concept, because they might not harbor specific mutations. Maybe you don't get the drug reimbursed, or the patient deteriorates too quickly. Taking all those aspects out, we look at the patients that have really received therapy, and you can appreciate that we do see an improvement of patients that have come from the progressive disease section in green and have actually provided an improvement in therapy. So we have an overall response rate of 47% when we include the mixed response in brown here. And there's, of course, a lot of data which is still in an analysis, because we do wait 3 and 6 months before we can judge response assessment. So again, this is an treatment concept which is heavily based on infrastructural aspects, such as the question of whether drugs are reimbursed, whether the payers are able to provide those drugs to the patients, whether the patients are fit enough for therapy. There's a lot of confounding factors. But I think you can see that once a relatively fit patient is entered into the program, it really can provide a benefit. Now, the question is, and this is shortly before I hand over to Michelle, is the question, how can we use data from the molecular tumor board to improve our patient, to improve our molecular understanding of cancer biology, and also, of course, for treatment. So we were focusing initially on lung cancer because it's still the cancer type with the highest rate of mortality and morbidity in the Western world. Projected cancer deaths until 2030, it is still way on top of the mortality score of all other compared cancer subtypes. So lung cancer is the most frequent cause of cancer death, and therefore we try to illustrate the benefit of studying genetic aberrations from our tumor board patients in lung cancer. Shown here are the genetic subtypes of lung cancer, of lung non-squamous NSCLC. You can see this here. I mean, this is standard knowledge. We do have a range of different mutations, which individually, such as ALK, BRAF, EGFR, occur relatively infrequent. Together make up about 20% of all cancers. We do have a lot of wild-type cancers in lung, and also some KRAS mutant, which we cannot target out of clinical trials so far. So what we did is we really identified our advanced stage lung cancer patients within the molecular tumor board. We performed a comprehensive genetic analysis within those patients and identified a genetic event within those lung cancer with a certain frequency, which prompted us to go further into studying this aberration with our partners in London. And this is the time now that I'm switching over to Michelle Dietzen. She's on the line now to present her part of this project from London, and I'm happy to then switch back once Michelle is done. So Michelle, all yours now. Thank you.
Thank you very much for this nice introduction. Yes, so I will talk to you today about how we are actually investigating cancer genome evolution and how this can then be used later for investigating different gene targets, which can then maybe be used for targeted therapy. So first of all, for people who might be not familiar with the term of heterogeneity, so there are different levels of heterogeneity in cancer. One level is the inter-tumor heterogeneity. So this basically describes differences between tumors. It can be with two different tumors within a patient or tumors between patients. But then when we zoom in further and we actually look at the tumor level, we can see different subgroups or different subclones of cells which represent different characteristics. And this level is called intra-tumor heterogeneity. And when we even go a little bit further and zoom into cells within the same subclone, we can identify differences between the cells. And this is the level of intercellular heterogeneity. Here in London, in our group that I'm working in, we are interested in analyzing intra-tumor heterogeneity, or in short, ITH. And for this, some really intelligent people came up with this great study called TracerX. For those people who are not familiar with it, it's a prospective observational study. We do high-depth multi-region whole exome sequencing. So this means we remove the tumor from the patients, and then instead of just taking one biopsy of the tumor, we actually dissect multiple regions Depending on how large the tumor is, we can get multiple numbers of regions. So smaller tumors will only have 2 to 3 regions, whereas some other tumors maybe even reach up to 5 regions. We have matched germline samples, and so far, so in total, the study will contain 842 patients. So far, we have successfully analyzed and published the first 100, and we're including only early stage non-small cell lung cancer patients. What do we do with the regions that we extracted and biopsied? So we run mutation and copy number analysis on it, and this can then help us. This can help us then to get an idea about when different events happened during the evolution of the tumor. So basically, when we compare the different mutations in the different regions and different copy number events, we can identify clonal mutations. So those are mutations that are present in all regions of the tumor. And we think that they happen early on. So they are present probably in the most recent common ancestors, and therefore in the trunk of the phylogenetic tree. Then subclonal mutations are mutations that are only present in a subset of the regions and therefore are represented in the branches of the phylogenetic tree. Here I show you the heterogeneous landscape of the different mutations in our cohort. So in total, we have 61 lung adenocarcinomas, 32 lung squamous cell carcinomas, and 7 other cancer subtypes in our 100 patients. And the top panel shows the total number of mutations within a patient, and the second panel underneath shows the proportions of clonal and subclonal mutations within a patient. What's quite nice, you can see on the top is we actually see a high variety of total number of mutations between patients, so that highlights the intertumor heterogeneity, so the difference between patients. But also when you look at the proportions of clonal and subclonal mutations, you can see quite a high diversity as well. So on the left side, for example, you see patients with quite a high fraction of subclonal mutations. So this means they have quite a high intratumor heterogeneity, whereas the ones on the right with the big blue bars have more clonal mutations and only a small fraction of mutations in their branches of their tree. The same what we apply here to mutations can also be done for copy number. So, for example, if we would look at a normal sample, so no cancer sample, we would assume that we see a total copy number of 2 all over the genome, like presented here in this panel. But this is not the case in cancer, and specifically not in lung cancer. Lung cancer is actually, one main characteristic is the high copy number instability, so really noisy profiles. If we have a look at one region, for example, of a tumor, you can see here we have lots of copy number gains and losses, or even quite prevalent in lung cancer, we can see whole genome doubling events, which leads into high ploidy tumors. If we compare now the profile of region 1 to other regions of the tumor, like for example here, 5 different other regions, on the first side they actually look quite similar, but to highlight that there are differences, I highlighted subclonal events in pink. So if you look at those pink boxes, you can actually see there are differences between the different regions. And this can be classified as subclonal copy number events. So we can identify the fraction of the genome with clonal and with subclonal events within a patient. If we add this to our overview plot, like it's done here, so the third panel down at the bottom now represents the fraction of clonal and subclonal events, but for copy number within our 100 cohort, you can see that there is actually also quite a high variety going on, and you can even see a slight correlation between the fraction of clonal copy number events and clonal mutations. One of the main findings that we had in the TracerX paper that was published 2017 in the New England Journal was that the subclonal fraction of copy number events, so a high subclonal copy number fraction, is related to poor prognosis for patients, which is very interesting. So copy number clonality, or copy number ITH, can actually be used as predictor for outcome. So the next step that we were thinking about is, but can we actually identify parts which are gained or lost more prevalent, so which are more targeted by different copy number events? So for this, we classified how frequently gained or lost our parts of the genome are. So here, for example, we look only at the lung adenocarcinoma, so at the 61 patients of the TRACERx cohort, and calculate the frequency of how often we see those different regions of the genome gained. But because of the multi-region purpose, we can actually classify between clonal and subclonal gain. So if I do this, so if I say, okay, clonal gain is the gain which is present in all regions of a tumor, we can get something like this. So in dark red, you can see now actually the clonal gains within those regions, and in light red, it's represented the subclonal gains. The same can be done for losses. So in blue, I represent the frequent losses within our cohort. And afterwards, we can also apply a significance threshold. This helps us to identify regions which are frequently significantly gained or lost within our cohort. One of the really interesting parts is chromosome 1q, where I also highlighted the gene MCL1, which is a pre-survival gene, which is prevalent gained in our cohort, and directly high on top of the highest peak within those regions, we identified MCL1. In total, 46 patients out of our 61 lung adenocarcinomas actually show a gain in MCL1, and 34 patients show a clonal gain, and 12 patients show a subclonal gain. What's quite interesting about the subclonal gain is that actually this means that the overall frequency of MCL1 gains might be underestimated, because if you imagine, if we only always take one biopsy of a tumor, but MCL1 is subclonally gained, so maybe in a different part of the tumor than the one that you just biopsied, you just miss this amplification of this gene. As the next part, we were then interested in seeing, okay, we see a significant gain of MCL1, but does it actually have an influence on the behavior of the tumor? So do we actually see a difference in expression for those genes? So does the gain actually have an influence? So for this, we compared the expression of the MCL1 gene in tumors where we see a gain of that part of the genome versus where we see no gain, and we can see a significant enrichment. So this helps us, or suggests that actually MCL1, or the gain of MCL1, makes a difference within the tumors. But one big part, or one big problem about those copy number driver genes, or that you try to identify, is in most of the time when you look at driver mutations, the genes are targeted by the different mutations. Whereas when you have a copy number event, most of the time it's not just the gene that's amplified, rather than it's a bigger chunk of the genome, or a larger segment, and it only includes the genes that might be selected for an increase or a decrease, or copy number gain or loss. So that's why we had a look at the segment size of the gained regions where MCL1 is located on. So here in this plot, I actually show the segments of chromosome 1q. And you can see on the top part, it's actually, lots of the time, it's a broad level gain. So you have, like, in the dotted line represents the position of MCL1, and you see it's actually a large chunk of the genome which is gained, and MCL1 is just located on it. But however, we were also able to identify focal gains, which are represented more on the bottom, which means even when you have like a smaller part of the genome or of chromosome 1 gained, it always seems to be around MCL1. However, we also don't want to say that MCL1 is the only gene that's driving this gain of 1q. So we also had a look at other genes that are located on this area, or on this significant gained region on the genome. And when we have a look at in how many cases, or in how many of those genes, we also see a difference in expression. We could even identify 316 genes, which also included 12 previously reported cancer genes. which highlights, like I said, that it's really difficult to identify the proper gene which is causing this. But however, because MCL1 seems to be, not just with our analysis, but also when we run GISTIC2, seems to be located on the highest peaks and prevalently lost in our cohort. And also looking at a pan-cancer TCGA data set, MCL1 is lost in lots of cancer types. So that's why we decided, okay, MCL1 might be a good target for treatment, and we want to pursue further analysis with this gene. So the next question that we had was, is MCL1 maybe associated to other cancer drivers? So we started looking at TP53, which is, like Philip already highlights, it's a very important cancer gene, and the mutation of TP53 is seen in lots of cancer types. So if we divide our cohort into TP53 wild type and into TP53 mutant patients and repeat the analysis of the frequency of copy number gains, you can see nicely here in this plot that the actual overall frequency of MCL1 gain is not very different between those two groups. However, you can see that in the TP53 mutant patients, you have a higher fraction of subclonal MCL1 gains, whereas in the TP53 wild types, it's mainly just clonal gains. Applying a chi-square test to this and to find out if this is actually a significant difference, and yes, we can confirm. So what we could find is that TP53 mutations are associated with subclonal MCL1 gains. We also had a look at other important driver genes containing EGFR and KRAS. However, we were not able to find the same association, so we couldn't find a difference between the clonality of MCL1 in KRAS or EGFR mutant patients versus wild type. So as a quick summary, multi-region sequencing gives us the opportunity to explore the clonal evolution of cancer. And what we've found so far with analyzing the TRACERx data, that lots of driver genes seem to happen early or clonal, so on the trunk of the trees of the patients. However, we also were able to identify lots of heterogeneous driver alterations, so which occur later or in the branches of the tree. And we could even identify that it's the case in 75% of the TRACERx tumors, of the 100 TRACERx cases. Then genome doubling and also ongoing chromosomal instability, so changes in clonal and subclonally, results in parallel evolution of different drivers. And one important bit that I already covered within my talk is we could identify that increase in copy number heterogeneity is associated with an increased risk of recurrence or death. Then in terms of MCL1, so we identified the pro-survival gene MCL1 frequently gained in our non-small cell lung cancer cohort in the 100 TRACERx patients. But like I said before, we also checked TCGA, and it's actually frequently gained in lots of other cancer types as well. And we could see a significant increase in expression in those tumors with an MCL1 gain. The high frequency of subclonal MCL1 gains actually suggests that the overall frequency of MCL1 gains might be underestimated. And we could identify that the subclonal MCL1 gains are associated with TP53 mutations. This is the end of my part of this talk. Thanks very much for having me and for letting me speak. And now Philip is going to continue with his part of the talk.
Michelle, thanks very much. You basically showed us that MCL1 is it's amplified in many cancers. And it might certainly provide a certain cancer vulnerability by inhibiting it. But the question really is, now that so many patients with lung cancer present with this alteration, and so many patients even in patients without lung cancer, other cancer types also present with this amplification of MCL1. What is this gene doing? Can we use it clinically? And what is its consequence for tumor progression? And therefore, I'm quickly showing you a few functional validation slides, which I consider very important to really utilize the data that we've generated from the molecular tumor board. Now, the TRACER-X data validated that it is frequent and very recurrent in lung cancer. And the question now really is, what's the consequence? You see here a very simplified signaling network present for programmed apoptosis or programmed cell death. MCL1 being a critical pro-survival protein in cancer cells, but also in physiologically normal cells. We know that a lot of MCL1 can protect cells against cellular stress. And, necessarily, a lot of MCL1, based on this amplification on the chromosome, might result in a very robust cancer cell. So what we did is we tried to utilize our knowledge to validate the frequency in other larger cancer cohorts. Using here is TCGA data showing that low-level gains or high-level gains occur in up to 36% of lung adenocarcinoma patients. And therefore, it is much more frequent than our conventional point mutants or mutations in oncogenic driver genes that we know from lung cancer. So we consider it a very frequent and recurrent alteration. And importantly, the genomic gain clearly correlates with an expression increase, shown here by the mRNA levels, comparing gain and no gain patients. You can clearly see that patients with a genomic gain have a clearly increased expression. Now we went on to study whether the increased expression would also result in more protein. We compare here normal tissue from an individual patient to its individual lung adenocarcinoma sample. So patient number 1, patient number 2, it's those on the left-hand side. You can see that the brown stain for MCL1 clearly shows you an upregulation of the MCL1 protein level as a consequence of increased mRNA expression. And this is also quantified on the right-hand side. So MCL1 is indeed overexpressed in lung adenocarcinoma. But what does it mean? Has it a functional relevance? So what we did is we generated a mouse model system in which we activate a KRAS mutant G12D endogenously in mice by infecting the mice with an adenovirus that carries Cre recombinase. The Cre recombinase deletes a stop cassette in front of the endogenous KRAS locus. We cross those mice into MCL1 floxed mice where we can co-delete MCL1 specifically within the endogenous tumor. We wait for many weeks until those mice develop an individual, their own lung cancers, and then we can study those lung cancers with or without MCL1 to understand whether MCL1 carries a functional relevance for those tumors. Indeed, what we see is that this is shown here by in situ RNA hybridization. We have a clear repression of MCL1 positive signal by using this model system. So it is functional, it works. And importantly, we can see that in this histology on the bottom here, where you can see this dark pink H&E staining for tumor lesions within those lungs, that we have a substantially and significantly reduced tumor burden upon loss of one or both alleles of MCL1, clearly indicating that MCL1 is important for tumor progression in those mice. Moreover, we checked for the level of MCL1 expression in KRAS mutant compared to KRAS and P53 tumors, and again observed here that upon loss of loss of P53, indeed expression level of MCL1, very much in line with what Michelle just said, are clearly increased. So we generated another mouse model system in which we not only activated mutant KRAS, but also deleted P53 in those mice. We wait for 6 to 8 weeks before we infect those mice, and then we treat those mice with pharmacological MCL1 inhibitors. So we really try to mimic a clinical treatment setting. Mice develop a tumor, get treated with an MCL1 inhibitor as a single agent, and then we follow them by CT scan over time to see how the tumor develops. And this is the 3D rendered image and the quantification of it. You can see that we can slow progression by treating those mice with an endogenous KRAS and P53 mutant tumor with an MCL1 inhibitor as single agent. And we really think that this is a substantial benefit by providing those animals with a fully blown multilocular lung adenocarcinoma with a reduced progression by just treating them with a single agent MCL1 inhibitors for only a few days. This is a proof of concept. It's of course not clinically translatable at this very moment, but it gives us the direction to consider MCL1 inhibition as a treatment concept in lung adenocarcinoma in the future. So with this, I'd like to summarize very briefly what we just talked about. We've seen that personalized oncology in patient care can instruct basic research in a true bedside to bench approach. So what we learn from the clinical department, we can transfer to the bench, and then of course we hope to get it back from the bench also to the bedside. We find that MCL1 gains, chromosomal gains, rank amongst the most common genetic events in pulmonary adenocarcinoma in patients as we speak, and we think it's one of the most common genetic event overall, certainly more frequent than the driver mutations that we care for in the standard treatment setting. We've seen that MCL1 is functionally relevant. It protects lung cancer cells against cell death, and it is important during tumor initiation, but also during tumor maintenance. And we therefore consider it a valuable drug target. From our work, we pose the hypothesis that a pharmacologic inhibition of MCL1 might serve as a novel treatment option for lung adenocarcinoma patients. Something that has to be studied clinically, of course. It's a long way to go, but I think the concept has been put forward here, and I'm very happy that we could generate all this data from our molecular tumor board approach. At this point, it remains me to thank a lot of funding agencies that have supported us over the last years, not only in Munich, but also in London. And it is a great pleasure that we had such a positive and proliferative interaction between Nicholas McGranahan's lab, Charlie Swanton's lab in London at UCL Cancer Institute, as well as the Francis Crick Cancer Institute, together with my group in Munich. And of course, we have transitioned now to the University of Graz, where we now perform our personalized oncology concepts, as well as our basic research concept in the context of the Medical University of Graz. I'd like to stop here. I'd like to thank you for your attention, and I think Michelle and me are very happy to take questions. Thank you.
Hello, this is Sonja Loges speaking. Can you hear me now?
Yes.
Very good. I'm very sorry for the beginning where I had apparently technical difficulties. So now I'm using the old-fashioned phone. So I'm very happy to moderate this webinar. I will not repeat now my introduction that you could not hear. I will just say that Michelle Dietzen is a very talented mathematician, data scientist in the third year of her PhD, clearly one of the rising stars in the field, and she works in the place to be lab of Charlie Swanton in London for the topics clonal evolution and the tracer X, which he has now presented to you. And then Philip Just, I know him for a very long time because we were together in a program of the German Cancer Aid. We both work on precision oncology, and I just want to say it's a big loss for Germany that he now went to Austria. still I congratulate him for his new position, and I hope that we can even work more closely together and maybe build some bridges between Germany and Austria. So now this is that, and I will ask some questions that were put forward by the audience. So the first one goes to Philip. Thank you both for sharing these concepts and data. I have a question regarding subclonal populations. In your experience, what allele frequency thresholds did you set for a particular mutation in order to consider it actionable? How were subclonal alterations taken into account when deciding treatments? Obviously, this would differ if examining tumor tissue versus liquid biopsy, but still curious about how your team approached this at the Molecular Tumor Board. Thank you.
So thanks very much for this question. I think it's a very pertinent and relevant question. There are two sides to it. On the one hand side, we do include a technical cutoff, which is put forward by the pathologists. They call a mutation only if it's 3% or above within their sample, which is a technical, as far as I understand, and I would have to refer back to the pathologists for more details here, but this is a technical measure. However, when we get this data then, and the mutation is reported in the Molecular Tumor Board, and it gets put forward into the Molecular Tumor Board, then the clinicians would look at it and judge the frequency of individual mutation within a cancer sample on two parameters. One is, what is the tumor cell ratio compared to normal cells within the sample? So how many malignant cells were within the sample, and how many stromal cells or bystander cells? In a pancreatic cancer, you would expect a lot of stromal compartment. In a lung cancer, you have a lot of tumor cells. And if you find let's say a 5% frequency of a mutation in a tumor that is very densely packed with tumor cells, you would need to consider it more differently than in a sample with a lot of stromal compartment. So as a rule of thumb, we consider everything above 5% clinically relevant for the individual tumor if the tumor cell burden within this individual sample was above 30%. But these are arbitrary measures, and I totally agree it's up to discussion of what the real and the best measure would be. But we try to make some functional relevance depending on tumor cell burden, really. And the second question was the subclonal events. We were not able to measure subclonality in our Molecular Tumor Board because we only have a one-shot sequencing NGS-based Illumina TSO 500 panel for every sample, really, and therefore we were unable to detect subclonality. Although I think it's a very important question and certainly interesting for a study in the future.
Yes, thank you, Philip, for this very eloquent answer. Then there's another question. I think this also goes to you. And I think the next question goes into a similar direction. So, sorry, I did not get it. Why is this not completely translatable into clinical practice? I think this question refers to the MCL1 inhibitor. And then the second question, which I think you can answer together, is what would be the timelines for the development of clinical bedside treatments in these personalized approaches?
So again, thank you very much for the questions. I think it's a very, very relevant aspect. Question number one was, why is it not translatable? It is, of course, we think it is translatable. We think it should be translated, but there's one caveat. And this caveat is that we observe in patients an MCL1 amplification, so an overexpression. In the mouse model, we study the deletion. So we basically look at a black and white mirror image of the situation. We try to judge the relevance of MCL1 by deleting it and therefore looking at the tumor development upon loss of this gene, whereas in cancers it is amplified. We try to basically do reverse genetics. We come from the backside to understand what it does, and therefore I think we still have to be cautious to overinterpret the data. I personally think that we should definitely try to use it in the first clinical setting to try and use MCL1 inhibitors, which are in in early phase clinical trials for myeloma, also in those patients, and it goes in line with the question of the timeline. I mean, timelines for getting this back into the clinical setting depends really very much on the question of how powerful your pharmaceutical support, industry support is. The study that we're talking about now took several years to complete, from the initial finding to the mouse model. So there is certainly a time frame of a few years attached to it, but we hope to get more rapid in the future by using in vivo CRISPR models where we can basically judge this much faster. Thank you.
Philip, maybe I can also ask a bit along these lines. Have you considered to use organoids or patient-derived xenografts and then utilize this inhibitor, because this would give you probably some more confidence if you choose patients with and without amplifications?
Again, a very good point. And in one of the figures in our paper that we've recently published on this topic, that you can look up, is organoid. We did an organoid model. So we did generate mouse lung cancer palmospheres, what they're called, so lung cancer organoids from KRAS P53 mutant tumors, deleted one or both alleles of MCL1, and indeed they do not grow in vitro as good. However, I also argue that those xenograft models and organoids only can represent a certain fraction of the in vivo situation due to the lack of stromal cells, immune cells, and so on and so forth. I mean, you know this better than I do, but I therefore think that going all the way into the clinical trial, I do see some benefit of using the actual in vivo endogenous lung cancer mouse model system. But again, it's a question of timing and a question of money, and those PDXs xenografts certainly do pose a certain relevant model. So I do agree on this.
Okay, thank you very much. Michelle, maybe I can also ask you something, because I think you probably have a different view according to your expertise on the MCL1 data. So if you when you think about it, do you think it is a specific type of amplification or something really special about MCL1? Maybe you can comment a bit on this.
So yeah, I think, like I said in my talk already, it's quite difficult to identify driver, like copy number drivers in different cancer types with genetics, because it is very difficult. You know, normally when you have a copy number event, it's not just the gene that's gained. But what I think was quite interesting about MCL1 is when we actually had a look at the sizes of the different segments in the different patients where MCL1 is located, we could actually identify quite a high frequency also of really focal gains just located around MCL1. And I think this helps, or this makes sense in terms of that. I think MCL1 is probably a really important gene for lung cancer, for lung cancers in general to develop and to get as aggressive as they are at the end. So I think MCL1 is probably a great target if we can get proper treatment for it. And I think it looks really promising if it can go back to the clinics.
Thank you very much. There's another question. I think this goes to Philip again. What is the proportion of actionable to non-actionable mutations in your data set?
So we, as I said, we do the TrueSight Oncology 500 gene panel, including translocations, RNA-based translocations. with this, we detect 500 most important cancer genes, hotspots. Some of the genes are sequenced more than just the hotspots; they are sequenced throughout. And we do have the data, and I have to be completely honest that I don't exactly know the ratio at the moment by heart. I would have to look that up, and I can certainly look that up. I can certainly say that the majority of mutations that we find is non-actionable. I mean, there are certainly more mutations that we do not have clear drug targets for. No, I mean, where we have clear drugs for, they are certainly known to be important, but we do not have clear drugs for it. This situation is improving as we get more availability of new drug classes as well. But I would have to look the exact number up. I think it's roughly about 30% actionable to 70% non-actionable, but I really wouldn't like to be too sure about this here.
Thank you very much. And then maybe I have another brief, I think, do we have to stop, or? Maybe I have one more brief question. So what are the structures? Because I'm not so super familiar with, for precision oncology now in Graz, is there a molecular tumor board? And who is sitting in this molecular tumor board?
So there is let's say a certain form of a molecular tumor board, the molecular Fallbesprechung. It's a concept which is basically centered around the very same concept of a conventional molecular tumor board. The question of which disciplines need to be involved to make a consensus decision in the molecular tumor board concept is developed as we speak here in Graz. We do have a very powerful molecular genetics and molecular pathology department, a very strong human genetics department, which are on the forefront of looking into personalized medicine concepts in cancer. And we have a relatively good accessibility of off-label drugs, prescribable, orally available off-label drugs. So this is a certain advantage to other sites, and we are in the process of structuring this into a conventional classic molecular tumor concept in the future.
Interesting. I'm sure you will also move a little bit more probably into liquid biopsy now that you are in Graz. Yeah. Okay, I think there are no further questions from the audience. Otherwise, all people in the audience can also raise the hand if they would like to say something or ask a question. Otherwise, I think we are going to close the webinar. Thank you to the speakers for this very fascinating presentations. And I think this is really a prime and rare example of translation, bench to bedside translation, and also a little bit back. I mean, and this is, I think Philip is really a role model for clinician scientists. So yeah, thank you very much. Thanks for all the questions. Have a nice afternoon and stay safe and healthy, and bye-bye.
Thank you very much. Thanks, Sonja.
Thank you. Bye.
Bye.